Ladies and gentlemen, we are starting with the session, and as has been advised by the organizers, half an hour presentation for each. Finish within 25 minutes would be better, so that we can have about 5 minutes for discussion. The first presentation, I think the topic itself is so interesting, integrating integrated plant breeding. In last few years, I have been witnessing a huge amount of progress in the genomics. But if you want to go into integrate the genomics in breeding, then you must have a very good phenotypic component as well. And over these years, while genomics has advanced, the phenomics is still, you find, is at the almost at the same level. Leave aside few examples where they have some kind of automation, but then the kind of complexities in phenotyping where you have P plus E put together, the E is going to come in, and there are various ways of circumventing it. One could be look for alleles of the genes which have very high heritability, so that the component of E becomes reduced. But in any case, as we move along, we are going to find solution to G plus P plus E, but at the moment we are with the E component, which remains most of the time unexplained most of the time in some of those traits which are of so importance to us. But we will go as along, we will learn as we proceed further. With these few words, I would like to, I have co-chairs Ms. Shobha, and I will take her help wherever it is needed. The first presentation is by Dr. Reif on hybrid breeding in wheat. And I will request Dr. Reif for the presentation.
30 minutes. Okay. Thank you very much for the kind introduction. Dear Rajiv, is he here? No? Ah, here, Rajiv. Thank you very much for the invitation. Okay, okay. It's a pleasure for me to be here in India to present today my talk about hybrid breeding in wheat. Does that does not work, the pointer? Can you? No, but I may. So maybe you can double check that. I think it's off. So technique is out of order, but it's no problem. So you all know that hybrid breeding is an unbelievable success story in several crops, such as maize, for instance. Yeah, and the reason for that success story is that in hybrids you exploit actually heterosis at a maximum way, and that leads to a dramatic yield increase compared to other variety types. And in addition, hybrids has the advantage to possess higher abiotic and biotic stress resistance, and at the end of the day you observe also a higher yield stability in hybrids compared to other varieties. While hybrid breeding has been successfully implemented in nearly most outcrossing species, it's still lagging behind in selfing species such as wheat or barley. And that stimulated us here to perform a public-private partnership project to investigate the potential for hybrid wheat breeding. And that was a joint project between 4 breeding companies and 2 research institutions. And the first thing we had to do is we had to search for the right material to go for hybrid production. That's not so easy in wheat. It's difficult to find good male lines in wheat, which are really outcrossers. Yeah? And at the end of the day, we came up with a list of 135 lines, 120 female lines. They are a little bit shorter. And then the male lines, they are a little bit taller and open flowering. And we did that. We selected those panel of lines using nearly all elite materials of the 4 breeding companies and conducting fingerprinting using microsatellite markers. And then our intention was to maximize the diversity in that panel of parental lines. We used then chemical hybridization agents to produce the F1 hybrids, so the single cross hybrids, and that is reflected here. So we crossed the 15 male lines with the 120 female lines, and surprisingly we could produce nearly out of 90% of the potential crosses enough hybrid seed for phenotyping. So that was quite excellent. So we applied genomics, that's clear, to that parental line. So we used first the 9K SNP chip, which was available at that time, and then later on the 90K SNP chip, and fingerprinted the set of 135 parental lines. And what I've plotted here is a heat map plot of the Rogers distances ranging from 0 to 1. So that means they are completely different, and 0 means that they share all alleles in common. And you observe here a very typical picture for wheat. You have no population structure present, a little bit of family structure, which is reflected here by these green boxes. And you have also no separation between the male and female lines. You see here in red branches, I marked the male lines, and here you will find a cluster of male lines here, but also here. So female and male lines are not clearly separated as that would be, for instance, in hybrid maize breeding programs. So we spent a lot of energy in that consortium to characterize this panel of 1,600 hybrids and their parental lines in multi-location field trials. We performed in total nearly 30,000 yield plots and 25,000 observation plots. So that's a massive phenotyping. And that paid off. At the end of the day, I plotted here the broad sense heritability only for the hybrids. It's a little bit higher in the inbred lines. And here in brackets, the number of location is given which was used for phenotyping. And for the most complex trait grain yield, we observed a high heritability of above 0.7. And you will see that we have also reached high heritability for the other classes of traits. So these are important economic traits for wheat. We had the opportunity to screen one abiotic stress resistance, frost tolerance. We have a range of different disease resistances, also very complex disease resistances like Fusarium septoria. And we observed also quality traits and realized quite a high heritability. So that was an excellent phenotypic base for our study. And first we wanted to investigate whether in wheat we have the same advantages as observed in maize. So we first tried to investigate the yield stability, contrasting this panel of 1,600 hybrids with the 135 parental lines using the stability variance. So if you observe higher values of the stability variance, the varieties are less yield stable. So the low values are desired. So then a variety type is more yield stable. And we observed for wheat a very clear picture that the yield stability was much higher and the stability variance was lower for the single cross hybrids in contrast to the parental inbred lines. So conclusion is we observe also the huge advantage of hybrids as compared to line varieties of a higher yield stability. Next point we addressed is whether hybrids possess also an increased abiotic and biotic stress resistance. What I plotted here is an index with equal weights of all biotic and abiotic stress traits we had observed. And this is the stress susceptibility, so negative values are desired. That means high resistance or tolerance against the abiotic and biotic stress resistances. And I plotted here the distribution of the lines in red and in gray of the hybrids. And you clearly can observe that the hybrids are on average more abiotic and biotic stress resistant as compared to their parental inbred lines. So, and now comes the most important points for the breeders. If you want to successfully sell your product, you need a higher yield in contrast to the other type of varieties. And this graph shows the grain yield in decitons per hectare of our hybrids in contrast to the top yielding varieties in German. So the highest yielding variety at the moment is this variety Tobak. and we observed a huge proportion of lines out-yielding this tobac variety, and I would coin that a commercial heterosis, and that commercial heterosis was in the range of nearly 10 decitons per hectares, and that was for the breeders quite convincing, so they thought that it was worth to spend so much energy in producing the hybrids and testing them in so much environments. So hybrid breeding in wheat has potential, yeah, that is clearly underlined by the phenotypic data which we collected. But the question is, with hybrids, that we have a huge number of parental lines available. So if you have a DH program running in wheat breeding programs, you can easily end up with 10,000 parental lines. And if you assume that you can use them as male and female, then you can calculate quite easily the number of potential single cross combinations you could produce out of 10,000 lines, and you end up with 50 millions. And that is even a too high number for the large breeding companies to test them all in field trials. So there is a strong demand in methods which allows you to select the right parents for hybrid breeding programs. And the most simple approach to do that is to look at the per se performance of the inbred lines. Most lines originate from inbred line breeding programs, so that means that their per se performance is well known. And then you could use the mid-parent value as a predictor of the hybrid performance. And what I plotted here in that graph is the correlation between the mid-parent performance and the hybrid performance, ranging from 0 to 1. And for the 13 traits which we have analyzed so far, and you can see that the mid-parent performance is quite a good predictor of the hybrid performance, especially for the less complex traits like quality traits or for these economic traits. You can really have good guesses out of the hybrid value looking at the parents. For frost tolerance, it was also quite high. For the diseases, it was also in an acceptable range. And the lowest value was observed, and this is also expected for grain yield. There you expect the highest degree of dominance, and therefore also the lowest correlation between the mid-parent and the F1 performance. So our question was then, what methods can we look at to improve the ability to select parental lines in hybrid wheat breeding programs? And you have heard in this conference already a lot of talks touching that topic of genomic selection. I nevertheless try to introduce that shortly, the principle of genomic selection. So you have a training population. It should be a quite diverse population, and this training population is phenotyped in intensive field trials. You have genotypic data available. You use these two sources of information to train a prediction model. And actually this is important that you don't select markers, but you use nearly all markers to train your prediction models. You come up with a prediction model, and then you have new untested genotypes which are then only genotyped, and you predict the values using your calibration model. So we use that genomic selection strategy, implementing the F-infinity metric, which allows us to tackle the additive and the dominance effects to predict the hybrid performance. And in order to evaluate our potential to predict the hybrid performance, we applied following cross-validation scenario. So you can see here again this vectorial cross of the 120 female lines and the 15 male lines. And now here below in that graph you can see how we performed the cross-validation. So we sampled out of these 120 female lines a subset of 80 parental lines, female lines, and 10 male lines, and formed the so-called estimation set consisting of 610 F1 hybrids. And these estimation sets have been used to train our prediction model. And then we used that prediction model to predict the phenotypic value of now different sets of hybrids. So we had always a remaining set of around 100 hybrids not exploited in the estimation set. So this is the most related test set in comparison to the estimation set. We coined that T2. So both parental lines have been evaluated in another hybrid combination. In the opposite is now this T2 scenario, a T0 scenario, where none of the parental lines have been evaluated in a hybrid combination in the estimation set. So this is shows the highest degree of divergence between estimation and test set. And we have something in between, this T1 scenario, where at least one female or one male line have been used in the estimation set to train our prediction model. So the range is T0, most far away, then T1, and T2 is the closest related one. And we applied that now in combination. I showed here the results for rich regression blob approach to evaluate the prediction accuracy. And these values have not been standardized by the square root of heritability, I have to mention. And you see here, this is the reference value, the midpoint value that we can reach, yeah, if you predict the hybrid performance using the midpoint performance. And this unrelated T0 scenario, we cannot improve our prediction accuracy with genomic selection, in contrast to the very simple approach of using midpoint performance. So if we have at least one parental line, or if we have one parental line tested in other hybrid combinations, prediction accuracy increases, which is visible here in this orange bar, and we really nearly reach the prediction accuracy of the scenario of the midpoint performance. And with the T2 scenario in for hybrid prediction, so we could beat the midpoint performance value and could do a better job applying genomic selection instead of using midpoint prediction. So I'm showing here at this graph the values for the other 12 traits, and this is depicted here, the relative increase in prediction accuracy, genomic selection versus midpoint performance. So if you reach a value of 0.1, so you have equal prediction accuracy applying genomic selection and or midpoint performance prediction. If you have value above 1, then you do a better job with genomic selection. If you have values below 1, you do not as good as using the midpoint performance. And the picture we have observed for cranial performance is now also the same, more or less, for all other traits. So in this T0 scenario, we never do as good job as using the midpoint performance. This is a little bit surprising, because for these easy inherited traits like leaf rust or stripe rust, powdery mildew, or the quality traits, we have expected a higher power of this genomic selection. And the T1 scenario, only a few traits really are better than the midpoint performance. And in the T2 scenario, we beat with genomic selection the midpoint prediction, and this is most pronounced in the first trait for grain yield. So what consequences can we draw out of this picture now for implementing hybrid wheat breeding? The first thing is there are traits here which I have presented which are sometimes difficult to phenotype. And for instance, frost stress resistance, that's not so easy. In Germany, for instance, this winter we have a lack of really a severe frost period which allows you to discriminate the phenotypic value of your panel of inbred lines. So that's very hard. So you have you can have no clue on that trait. And there it's definitely of advantage to using this kind of methodology. And what I've shown you here is the contrast of using a kind of marker-assisted approach. So we applied association mapping, exploiting additive effect exclusively or dominance. And this first bar is exploiting additive and dominance effects together. And then I plotted here two genomic selection approaches, BASE-Pi and R-BLUB. And you can see that we reach this accuracy of 0.6. So this is of a value which is better compared to having no idea on how good your lines are performing for that trait which you may not phenotype at a certain year. So, but this is for most traits you have a very reliable phenotypic basis every year, and then the question is really that you have to stick to this scenario which makes sense, where you can really do a better job than using the midpoint performance values, and this is this T2 scenario. And if you would apply this T2 scenario, you could end up with a kind of mating design, an unbalanced or balanced missing factorial mating design, which is plotted here, so that you only produce a part of these hybrids which are marked here in these green boxes, and that you don't produce, because it's very cost intensive and very difficult also in wheat, that you don't produce all hybrids, but that you predict them, and then you are still in this T2 scenario, and you can do a quite good job in predicting the hybrid performance. So that might be one approach to implement genomic selection in hybrid wheat breeding. Okay, I want to use the remaining minutes, and I can see that I'm still good in time. Now shifting away from the potential of genomic selection towards marker-assisted selection to illustrate you a little bit the benefits of using hybrid breeding, where you work with two gametes at the same time. And what I have plotted here is quantile plots of the p-values, so that illustrates that we used an association mapping approach in this hybrid population, which allows us to map additive and dominance effects, implementing this F-infinity metric. It's like using an input line panel, so you have to correct for family structure, and you can do that by implementing a kinship matrix for the GCA effects. And if you apply this kind of association mapping approach now in this panel of 1,600 hybrids, so you can try to first map genes or QTLs, and then in the second step you can try to estimate the additive and the dominance effects, and we did so. And I presented here the results for the leaf rust, for the trait leaf rust, where we detected 4 major QTLs, and every QTL explained at least 10% of the phenotypic variation. And I used the presentation, which you may be familiar, for instance, which is also used in the famous book of Falcon and McKay. I plotted here the 2 homozygous genotypes, so this is the resistant one and this is the susceptible one, and then you have the mean, and you have this A effect, the additive effect, and then you can plot also the estimated dominance effects. And looking at that pictures, you see that we have for all 4 major QTLs a degree of dominance which is close in the range of complete dominance. Yeah? And that is quite an important benefit for hybrid breeding now, because you have to focus for this disease resistance management only in one pool. Yeah? And you can fix the resistance now in one pool and exploit that degree of dominance then in the hybrids. And to illustrate that potential of this disease management by exploiting dominance effects, I presented here a box whisker plots contrasting now the input lines and the hybrids for the different genotypes for the 4 detected major QTLs. And I want you to focus now on that red-filled box whisker plot, where these first 3 resistant genes are fixed now for that type of line varieties. And you can see here a very low susceptibility, leaf rust susceptibility. And you can contrast now this class, which would be typical for line breeding, that you have these alleles in the homozygous state, now with a class of hybrids where you have these 3 resistant genes, the first 3 in a heterozygous stage, and you observe now nearly the same mean value, which reflects the potential really to have an easier handling of disease resistant management using hybrid breeding instead of inbred line breeding. So, good in time to summarize my results. I hope that I can convince you that hybrid wheat breeding is a promising approach to increase yield, yield stability, and also abiotic and biotic stress resistance. Genomic selection is a valuable tool to predict hybrid performance depending on the relatedness between training and prediction population. And marker-assisted selection enables sophisticated approach to exploit dominance effects in these hybrid wheat breeding programs. And I want to thank all the partners or the 4 breeding companies who did an excellent job in producing the phenotypic data and the genomics data. I want to thank my colleagues from the university. No, I was too quick. I want to thank my colleagues from the University of Hohenheim, especially Friedrich Longin and Manje Gouda. And I want to thank my colleagues at the IPK, Yushin and Florian, for their excellent work. And I want to thank you for your attention, and I'm happy to answer your questions.
So yeah, just following up on the dominance question, because that was one thing I was thinking about when I heard your presentation. So could you comment about what, in your prediction model, you had one variance component for the additive and one for the dominance, right? What would be the ratios between those 2 variance components? On the one hand, like you showed some results for those specific genes where you see that there's a strong dominance, but I'm wondering, on a genome-wide basis, does the dominance not contribute to the predictions then? Is that what you're saying?
Yes, at the end of the day, yes. So it's trait dependent, so this is the point I wanted to make. So for granular, you can't do a job. So actually, it's much more difficult to precisely estimate also these interaction effects, or the intralocus interaction effects, the dominance effects. That is one explanation. You have a positive correlation, yeah, but it's still associated with so much noise that you can't do a better job for granular, yeah. But there are traits where it's very important to model, especially for these areas where you have large effects, where you model in the dominance properly.
I might be asking roughly the same question that Aaron asked, but it seems to me for T2, if you want to estimate or have an idea of the additional efficacy of marker-based prediction over a baseline of pedigree information, the proper comparison is not mid-parent, but line blobs. Did you look at that from the hybrids?
What is in your opinion the reference base? The general combining activity effects? Or?
Well, the effect of a line, the blob of a line. Yeah.
So that means
GCA.
GCA effects. Yeah.
Did you make that contrast?
We have not yet investigated the contrast in that cross-validation scenario. So we leave one out, yeah, and then it's not so positive. And you have to think in one point, and this is very hard to understand for a maize breeder. Yeah, for maize, it's very easy to implement top-cross tests. Yeah, from the technical viewpoint, it's very easy. For wheat, it's nearly impossible. Yeah, so that's not attractive for a wheat breeder to implement top-cross tests at the first stage. They would immediately go for hybrids. So that might be important information.
In any hybrid breeding program, we need a stability of hybrids. So one is we need to test across multi-environment. So it may take, in the initial stage, we may not be able to test in large environments. We start with few environments and go to more levels of environments. Does prediction model also takes into account stability of performance in 1 year? With 1 year prediction data, can we say that hybrid is stable for many locations?
So this is, here actually we don't model genotype times environment interaction effects. That is a wheat breeding program in Germany, and you can exploit that hybrids then in Germany. It's very hard to transfer these type of varieties to France or UK due to quality issues. Yeah, so bred quality is different in Germany than in France and in UK. Don't ask me about the ranking. Yeah, and that makes it very difficult to be focused on that target environment, Germany, so it does not make sense to model genotype times environment interaction effects. With regard to taking that into consideration in this genomic selection part, so what I presented here are the values not standardized with the square root of heritability. So, and I think it was mentioned in earlier talks, that the heritability is a very important measure. So the square root of heritability is the maximum you could reach, yeah, in your prediction approaches. But at the end of the day, this is only you have to implement that in your breeding programs. Yeah? I'm not a fan of having your training populations outside of everything, phenotype them intensively, but I believe that this must be integrated in the breeding population, and the breeding process, and if you do so, you have to be well aware that you will never reach that value, the correlation standardized by the square root of heritability, but actually that you have to live with the correlation between observed and predicted values.
Well, thank you.
So this is, yeah? Pardon? So you asked me about the...
Chemical hybridizing agent.
It's a product from Saatenunion, Croissoir, which has been used.
Which one? Sorry?
It's Croissoir. It's a Croissoir. It's a chemical, syntophen is, I think, the chemical substance behind that. I can show you later. I'll give you the reference of that. Yeah, and it's close to be released in France and European-wide.
Well, we've gone a little bit over time. Thank you very much for the questions, and thank you very much for the talk, Dr. Reif. And